Okay, so good morning, everyone. I am Hasin Rehana. I'm a PhD student at University of North Dakota. Today, I will be presenting our research on automated extraction and classification of vaccine adjuvant mechanism using large language models. Adjuvants are critical components for vaccines. They don't actually act as antigens themselves, but they make immunoresponse stronger, faster, and long-lasting. Each adjuvant operates through specific mechanism of actions, such as activating the innate immunopathways or modulating T cell responses. Understanding those mechanisms is the key to design the next generation safer and more effective vaccines. As of today, several databases exist. For example, VaxGeo, AdjuvaDB, Vaccine Adjuvant Compendium, which curates hundreds of adjuvants. Those resources are obviously invaluable, but they rely heavily on manual curations. As the literature expands rapidly these days, it became extremely difficult for the experts to manually track and classify all those mechanism informations. So our motivation was to ask, can we use LLMs to accelerate this process and assist the expert curators? But there are few challenges that make these automations difficult. For example, the language in the immunology papers is highly technical and nuanced, often describing mechanisms in complex and indirect ways. Secondly, we see a lot of variations in the adjuvant names and formulations. The same compound may appear under multiple synonyms or preparation methods. And third, ambiguous terminology. Terms like stimulation or activation can mean very different things depending on the context. So our objective is to automate the extraction of adjuvant names and immunoresponse mechanisms from the literature, analyze patterns, linking adjuvant types, mechanisms, and outcomes, and develop a curator co-pilot to reduce the manual curation bottlenecks and ensure consistent mechanistic annotation. Here is our overall workflow diagram. We start with the PubMed abstracts, and we use LLM in two phases. In the first phase, the LLM identifies the adjuvants and extracts corresponding immunoresponse mechanisms from the abstract. And then in the intermediate phase, they are grouped by adjuvant names. And after that, in second phase, LLM summarizes and organizes those mechanism descriptions for each unique adjuvants. Once we have done the LLM part, then we compared the extracted results with the existing VaxGeo database to identify novel or missing adjuvants, followed by expert manual validation. This human-in-loop design ensures both scalability and reliability. So basically in phase one, we focused on entity extraction, capturing pairs like activates the NLRP3 inflammasome. And in second phase, we used summarization and classification to refine those into structured mechanism summaries. So each output looks something like this. This structure actually makes the data machine readable and ready for the next phase for the ontology integration. This is a sample final output. For LPS, the system summarizes that it activates the TLR4 pathway, which triggers the dendritic cell activation and promotes the Th1 responses, all supported with PubMed IDs for traceability by the manual validator. So across around 800 abstracts, we identified a diverse set of mechanisms. T cell activation and dendritic cell activation together accounted for over 1/3 of the extracted mechanisms. TLR4 signaling appeared about 14% of the cases, reflecting its central role in innate immune sensing. We also observed frequent mention of B cell antibody production, pattern recognition, receptor sensing, cytokine signaling, which aligns with known immunological cascades. We also examined how the mechanisms co-occurred. Interestingly, dendritic cell activation, T cell polarization, B cell antibody production, those commonly occurred across many adjuvants, suggesting that synergistic engagement of multiple pathways may define highly effective adjuvants. Certain mechanisms like NLRP3 inflammasome activation were much more specialized, tied to very specific adjuvants such as ALM or CpG formulation. To test real-world utility, we tested this pipeline as a curation co-pilot framework that screens literature against Baxter database. The result was encouraging. The system flagged around 27 adjuvants that were not previously captured by the Baxter database. This demonstrates that the pipeline can serve as a practical human-in-loop co-pilot, helping curators to stay up to date as the literature evolves rapidly. So this slide shows a mock version of what a curation copilot interface could look like in future. Here, a curator can upload a file consisting the PubMed IDs or run query directly here and download the PubMed abstracts and then apply LLM in different phases by their customized query, or we can also have a default query set. And then it will automatically compare with the existing database, and a review queue will be generated along with the evidence. Experts can then validate, edit, or export entries directly to Baxter, keeping the human firmly in charge, but with LLM doing the heavy lifting behind the scenes. So in summary, we developed a scalable LLM-based pipeline for automated extraction and categorization of vaccine adjuvant immunomechanism. It functions as a curation co-pilot, assisting expert reviewers and improving consistency of mechanistic annotation. It has discovered 27 novel adjuvants that were not previously captured by Baxter database, which is very promising. And it actually transforms the unstructured biomedical literature into structured, actionable knowledge insight from rational vaccine design. So looking forward, we aim to build a user-friendly end-to-end copilot for community use by integrating dynamic ontology support and domain-tuned biomedical LLMs. Our long-term vision is to use this AI system that empowers the experts rather than replacing them, accelerating the discovery across immunology and vaccine research. I want to thank and acknowledge my collaborators from University of North Dakota, University of Michigan, and Bukowski University, as well as the HARLab members and our funding from NIH. Thank you all for your attention. I look forward to your questions.
Hey, thank you, Hasin, very much for the excellent talk. All right, so let's open it up for questions.
So, Hasin, yes, great talk. I have a question about your like a curation co-pilot one. You said it's a mock version. Is the co-pilot project the one you are using for your project, or it's more like your future work?
This is like my future work. So all those parts like were done for this test, but they were like different components. But in future, I want to integrate them to a end-to-end copilot, and it can be done using a web system. Yeah, but this one is just a mock version.
I see. So if in this case, do you also need to put your prompt into the structure, or you don't need?
So my plan is to have a default prompt, but also having a edit option that so that the curator can like tweak the prompt based on the specific need of the project. And
Okay.
also we can have a like follow-up question part, but that is just an idea. Yeah. Thank you so much for your question.
Okay. Thank you.
Great. Great.